At the microscopic level, human conception is a fiercely competitive mechanical process. Millions of gametes are deployed, but only one successfully breaches the zona pellucida of the ovum to initiate a new biological timeline. But scale that biology up to the level of public health, and human reproduction becomes something actively managed. This photograph from mid-century India shows an early family planning initiative, marking the historical moment when governments realized that unmanaged population growth outpaces medical and economic infrastructure. To manage this on a clinical level, modern medicine does not rely on chance. Instead, it targets precise physiological checkpoints along a strict chronological timeline. This intervention matrix rests on three clinical pillars: preventing conception, managing or terminating gestations, and engineering conception when biological systems fail. Every decision made within these three pillars is strictly bounded by the patient's anatomical constraints, the specific etiology of an invading pathogen, and rigid gestational timelines. Reproductive medicine is a flowchart. It maps biological roadblocks and builds highly engineered bypasses to achieve or prevent a viable pregnancy. We begin with preconception prevention. Surgical contraception, specifically vasectomy in males and tubectomy in females, is the definitive mechanical block against fertilization. This schematic of the reproductive tracts shows how the block is applied. By severing the vas deferens or fallopian tubes, the surgery isolates the gametes. Inside the testes and ovaries, gametogenesis continues uninterrupted. Sperm and ova are produced. They simply cannot travel. Because the endocrine system remains fully intact, the hormones driving this process, testosterone and estrogen, continue to be synthesized and released into the bloodstream. Normal physiological drives and secondary sexual characteristics are entirely unaffected. Surgical contraception provides a highly effective, though poorly reversible, mechanical intervention. It neutralizes the transport mechanism while leaving the underlying cellular biology fully operational. When we shift from mechanical barriers to pathological threats, the focus turns to the specific etiology of sexually transmitted diseases. STDs are strictly categorized by the biology of the pathogen. Bacterial and protozoan infections are curable. If detected early, antibiotics can completely eradicate the invading organism. This electron micrograph shows the spiral-shaped Treponema pallidum bacteria, responsible for syphilis. Like gonorrhea and chlamydia, it presents a physical structure that medicine can destroy. Viral STDs, however, operate through a different mechanism and represent the critical exception to curability. This image highlights small yellow viral particles deeply integrated into the surface of a host cell. This is how HIV, hepatitis B, and herpes behave. They hijack the host's genetic machinery, resulting in a lifelong, incurable infection. A pathogen's biological classification dictates whether an STD is a temporary infection or an incurable disease. This is why mechanical barrier methods remain the primary prophylactic defense. Once conception occurs, the clinical timeline dictates the options. Medical termination of pregnancy, or MTP, is governed by strict physiological and gestational constraints. This timeline graphs gestational development. During the first trimester, up to 12 weeks, MTP is clinically safe, requiring approval from a single practitioner. Between 12 and 24 weeks, escalating maternal risk mandates a strict two-practitioner approval protocol. This exact same gestational timeline is used for diagnostic screening, primarily through a procedure called amniocentesis. The mechanism relies on extracting amniotic fluid from the uterus. This fluid contains discarded fetal cells, which are then isolated in a lab for karyotyping, a visual mapping of the fetus's chromosomes. Karyotyping detects chromosomal abnormalities, revealing conditions like Down syndrome or hemophilia. It cannot detect metabolic diseases like diabetes, which do not present as missing or extra chromosomes. Because karyotyping also reveals the sex chromosomes, there are strict statutory bans on using amniocentesis for sex determination. Post-conception interventions require diagnostic targeting and absolute adherence to gestational timelines to balance maternal safety with clinical ethics. When conception fails naturally for over 2 years of unprotected intercourse, a couple is clinically diagnosed with infertility, triggering the deployment of assisted reproductive technologies, or ART. This split-screen model contrasts in vitro lab procedures with in vivo anatomy. In ICSI, a microneedle injects a sperm into an ovum. Zygotes with 8 or fewer cells undergo ZIFT, placed into the fallopian tube. More developed embryos bypass the tubes via intrauterine transfer. In vitro techniques are not arbitrary. The specific anatomical destination is dictated entirely by the cellular development stage, or blastomere count, of the embryo being transferred. Shifting to in vivo processes, the body's internal architecture is utilized for fertilization. In GIFT, gamete intrafallopian transfer, harvested ova and sperm are placed directly into the fallopian tube of a female unable to produce eggs, maintaining a functional reproductive environment. The absolute anatomical prerequisite for GIFT is structural integrity. The fallopian tubes must be unblocked for the gametes to travel and implant naturally. For cases of male impotency or low sperm count, artificial insemination and IUI, intrauterine insemination, are utilized. Sperm is introduced directly into the vagina or deeper into the uterus, mechanically bridging the transport gap. In vivo techniques succeed by leveraging the patient's healthy anatomical structures, bypassing isolated gamete failures, while relying on the body's natural physiological processes. When we pull back from the microscopic details, the macroscopic matrix of reproductive medicine becomes clear. It is the synchronized management of preventing conception, managing gestation, and engineering conception. Clinical application is an exercise in precise deduction. It requires locating the exact failure point, whether an incurable viral pathogen, a surgically blocked transport tube, or an insufficient sperm count. The alphabet soup of treatments, MTP, ICSI, ZIFT, GIFT, only functions when strictly mapped against the body's biological constraints and rigid chronological timelines. Modern reproductive health is a highly engineered system of targeted workarounds designed specifically to navigate and triumph over mechanical, pathogenic, and cellular failures.